Okay, and welcome to the pioneer session of this afternoon. It will be the last session. It will be chaired by Andrea and by myself. And Andrea and I, we are very proud that we can introduce you our three pioneers of echolocation research. And these three people, Alan Grinnell, Jim Simmons, and Uli Schnitzler, did some really groundbreaking research in the last decades, and they still are active and do ongoing research. We changed the structure of this session a little bit, so we will start by both of us will start introducing the three pioneers, and then the three pioneers will give their talks on different topics. And at the end, we will ask them to join us here in the front, and we will have a sort of a discussion, a roundtable talk, and we would invite you to ask questions and maybe leave some of the questions to the talk for this last part of our session. And yeah, now I will give you the stage.
Okay, so I have the honor of introducing and talking about Alan. So as an undergraduate student at Harvard, Alan walked into Don Griffin's lab and said, "Do you need an unpaid research assistant?" And Don said, "Yeah." And so the rest is history. So Alan started his work in echolocation, as I said, as an undergraduate student. And his first project was actually this super interesting project looking at how the bat changed the interpulse interval of its echolocation pulses as it tried to avoid wires of various diameters. We've heard a lot in this session about how changes in interpulse intervals are a metric of task difficulty for bats, and this work was actually first done by Alan in 1958. Alan and Don then went on to do what really remains the best quantitative study of the resistance of bats to jamming. For his doctoral dissertation, Alan was the first to study the neural bases of echolocation in bats. He studied two bats, the little brown bat and Townsend's bat. And I put, I knew I was going to do that. And you could see that I put the names, the titles of all of the, really extensive, lengthy papers that came out of his dissertation. Just some highlights. He showed that evoked potentials and single neurons and the colliculus respond to ultrasonic signals. So that's really important. He showed single neurons had very sharp tuning curves, also very important. He showed that the colliculus responds to changes in the direction, both vertical direction and horizontal direction, ultrasonic signals. And this is actually my favorite result of all of this work. He identified neurons that he said had time measuring capabilities. And if you're an echolocating bat, you need neurons with very, very sharp time measuring abilities. And just to show in this slide, the experiment was to present two tones or two FM sweeps, vary the time interval between them, and look at the interval at which the response to the second tone reached 100% amplitude of the response to the first tone, or even higher. And I knew I was going to do that. And you can see I circled here that in a time interval of 0.5 to 2 to 3 milliseconds, you're at 100% recovery. So and that's really an amazing finding. So not only did Alan discuss, no, I got it, study neural bases in those two species of bats for his dissertation, he went on to study multiple bats, 7 New Guinea species, several neurotropical species. And what is really exciting about this work, and I showed the graph here, Alan was the first to actually show that the mustache bat has an acoustic fovea, which is an amazing result for echolocation research. But Alan, as I said, went to New Guinea and went to Panama, and he has continued to study bats all over the world with Uli Schnitzler and with Don Griffin and with Jim Simmons. And these are just some of the very many titles of work that he has done in the field, looking at echolocation behavior of various species of bats. And before I go on, Alan told me, one fact which I didn't know today, and I feel embarrassed because I spent a lot of time preparing for this talk, is that in 1967 he was in a group that was actually the first to record evoked potentials from the dolphin's brain. And what they showed in that work is that the evoked potentials in echolocating dolphins also show the fast recovery times as done in bats. And they also verified this, what was only a speculation up to that point, that dolphins can hear sounds through their lower jaw. So really, really quite remarkable results. And let's see. And then I will turn it over to Annette.
So now let's step back to the year 1966, when in Frascati, the first international animal sonar system took place. And here you see a lot of the early pioneers like Don Griffin, who discovered echolocation, Fred Webster, Nobusuga, Al Novik, and also Alan Grinnell here in front of the column. And this was also the meeting when Uli Schnitzler entered the stage. It was his first meeting, and it was also the first time that Alan and Uli met. And since then, they have a very close friendship. So Uli, after serving in the Navy, studied biology in Tübingen and started his PhD with Franz Peter Moeres. And in Frascati, he presented his first data. So he did an obstacle avoidance experiment in horseshoe bats. And so So he trained the bats to fly from a starting place through a frame with vertical wires to land on a landing bar. And at that time, there were no tape recorders available and also no fancy sound analysis tools as we all have today. So he recorded the echolocation signals with a microphone here, and he heterodyned the signal with the resting frequency of this individual's bat. So you have to know these bats emit constant frequency signal, of course, with an FM at the end. And then he also had the flight speed from 2 photocells, and then he measured the sound parameters in this heterodyne time signal of the call. And he already noticed that there is an offset between 0 and a deviation from 0. And he estimated that this is approximately the Doppler shift when the bat's fly towards the microphone. So then he went to Frascati and he presented his first data. And then when he came back, he accidentally adjusted the heterodyne frequency higher than the resting frequency. And he suddenly noticed it's not an increase in frequency, but a drop in frequency. And he was totally confused. And it gave him several sleepless nights questioning his research until he found the solution. It's the bats that do this. And this was the moment when he discovered Doppler shift compensation. So the bats emit a constant frequency call here when they hang, and immediately when they start, they lower the emission frequency. This is when they land, so that the echo frequency is kept at the so-called reference frequency, which is the frequency that fits best into the auditory fovea, acoustic fovea, Alan basically discovered. So after this, after his PhD, he went to become a postdoc in Griffin's lab, where he also studied Doppler shift compensation in the wind channel, and he proved Doppler shift compensation in Pteronodus parnellii. And then he did, he went back to Tübingen, where he did his habilitation. He became an assistant professor in Frankfurt and found a fuller professor in Marburg. And in this time, there was also Alan visited his lab in Frankfurt, and they measured the directionality of the emitted call of a horseshoe bat, and also the directionality of hearing by using the Doppler shift compensation behavior of these bats. And up to now, this is the only contour map of a, contour map of a bat, which shows both the directionality and the direction of the sound emission, and combines both the direction of sound emission and of hearing. He also started to do electrophysiology, measured behavioral audiograms, cochlear microphonics, auditory nerve responses, and also proved Doppler shift compensation in hyposideroids at that time. And he published several reviews. And this review here, this is the 1980 review. Is a really recommendable review because it summarizes all the knowledge and all the literature from the beginning of echolocation research until 1900. He showed that each insect species creates its own glint pattern on the carrier frequency of the echo, and that the flutter detecting forager use these glint patterns to classify prey even independent of aspect angle. And he also did electrophysiological studies together with Jo Ostwald, and he showed different response pattern of cortical neurons to the natural echoes from fluttering insects with different wing beat rates and aspect angles. And again, he wrote several reviews, and the latest, the auditory phobia and Doppler shift compensation, is from 2011, and it combines or summarizes all our knowledge on Doppler shift compensation in flutter detecting foragers. And even today, he's still active doing research in the lab on Doppler shift compensation, which you can see on this article only published recently. So, but flutter detecting foragers was not the only research field in which he was active. In the 1980s and 1990s, he investigated the performance of echolocation systems and the auditory information processing in psychophysical experiments. Motivated and really challenged by Jim Simmons' publications, he put much effort to solve the questions how bats process information in the echoes and which receiver type may be implemented in a bat's brain. So this was a discussion that dominated the whole field for many, many years. And the discussions were sometimes tough, sometimes emotional, but always exciting and always fruitful. So he summarized the results from different labs together with Dieter Menny and Heidi Hackbert in 1987, and with Cindy in 1998, where they summarized the results from all these behavioral studies in the lab And they also summarize the critical discussion on this topic. In his own opinion, his personally large milestone to understand the echolocation systems of bats was when he started in the mid-1990s to study bats in their natural environment together with Elisabeth Kalko and later also with me. This opened his eyes for the diversity of this group, and in a comparative approach, he addressed the question how the motor and sensory systems of bats are adapted to where they forage, how they acquire their food, and how they find their food. So he defined different habitat types according to the distance of the prey to the background, which you see here, open space, edge space, and narrow space. And he also showed that bats hunting in edge space reduce the sound duration when they approach the background to keep the overlap window open so that the prey echo is not masked. This, his own studies, but also the publication of many researchers in ecology of single species finally led to the guild concept, which attributes species that perform similar tasks and share similar adaptations into functional groups. But this was not everything. So when he started in Tübingen, he established a completely different field on acoustic startle response and the mediation. I won't talk about this, but it was a complete field, and about the same number of students and postdocs worked in this field as in echolocation. So in 2008, I have to say, he retired officially, but immediately became the first senior professor at the university in Tübingen. Also in 2008, he started a very fruitful and ongoing cooperation with Wu Dinh Tong from the Institute of the Vietnam Academy of Natural Sciences and Technologies, and he studied there, is still studying the Southeast Asian rhinolophids and hippociderids. Uli is in a fortunate position to have all freedom to do whatever he wants without annoying duties of having a university position. He's still very active. He is a brilliant and analytical mind, and it's fun to do research together with him and discuss research with him. So, Uli, we are very much looking forward to your talk with your newest perspectives on doppelshift compensation after having spent, I don't know, hours flying on the back of a bat through Bulgaria. So we will hear about this later.
Okay, so Jim Simmons started studying bats as a graduate student in psychology at Princeton University, where he started working with a great comparative auditory neuroanatomist and physiologist, Glenn Weaver. Glenn Weaver is actually very close to my heart because he still has the definitive book on the amphibian ear. So Jim began studying, his idea was to train bats in a laboratory environment to get an idea of their acuity of hearing and the acuity of the echolocation system. And what he did was to modify the Lashley jumping stand that was used to develop to study visual discrimination learning in rats with both flying and later on, as you'll see, with stationary bats. And here's Jim in his very younger days training a bat, and you can see the size of the equipment, much of which is still in the lab today. We're trying to get rid of it. Some of which is still in use. It's quite amazing. And Cindy in her presentation talked about some of the results. And so the initial results from the initial experiments is that big brown bats were able to discriminate by psychometric function using an arbitrary criterion of 75% correct, difference in targets of about 60 centimeters, which difference in targets of 60 centimeters, which is a difference of about 60 microseconds. Well, if people thought 60 microseconds was bad, Jim moved to Washington University, where he took his first job as an assistant professor, and he was very pleased to tell people all those years at Washington University, he taught introductory psychology. So those students learned a lot about bats as well as about other issues of psychology. So in these experiments, using better equipment, better delay lines, and working harder to train the bats properly, he found that bats were able to discriminate differences between two targets of 1 microsecond, so 1 microsecond difference. If that wasn't bad enough, he showed in a 1978 paper in Science is that the bat's performance, the bat's performance follows both the fine structure and the envelope of the autocorrelation function of the signal, which means that they're operating as a fully coherent receiver. Not only that, but you can see that the bat shows more errors at the side lobe of the autocorrelation function, which is an area of phase ambiguity, which indicates that the bats may be able to detect the phase of the ultrasonic signal. So after teaching introductory psychology at Washington University, this is a 1973 headline in the student newspaper: psychologist studies bats SODAR, as opposed to the things that psychologists should study, like human behavior. Okay, this work was controversial. What was more controversial is when Jim moved to Brown University in 1984 and started these experiments with students at Brown, and again, using better delay lines in the experiments, they found that under some conditions, bats, and you think I would learn. The psychometric functions show that under some conditions, bats may be able to detect differences in target distance of 10 nanoseconds. And as Cindy pointed out, and Meta pointed out, this was highly controversial in the field. It remains highly controversial in the field. This is a picture of everybody in 1994, where Jim signed the Declaration of Sandborg, where he admitted that the data would only be correct, the 40 nanosecond value, discrimination value would only be correct if the bat was able to compensate for all of its movements. Now remember, the bat's not fixed, it's on a two-choice platform. And it can move, and it can move its head, and it can move its ears. And you would only get that 40 nanosecond threshold if the bat was compensating for its own movements. But I really want to point out, and I know this is controversial, everybody's smiling. Everybody is smiling here. So although these were intense discussions, they were not hostile discussions. And I think that shows a lot about the personality of Uli and the personality of Jim, as well as Cindy. For those of you who don't know Lee Miller, everybody was very respectful and very friendly with each other. Okay, so moving on. So when Jim moved to Brown, he looked, the research he first moved into was not looking at discrimination thresholds in a two-choice task, where the bat has, one echo compared to another echo that's varying in some way. The question here was to look at the bat's ability to resolve individual glints and echoes. So bat sends out a pulse, the pulse insonifies, say, an insect, and it comes back different from the pulse because it's hit on something in the environment. And the differences are frequency peaks and nulls, which we call glints. And in this experiment, again, with Cindy Moss, you could see, again, the psychophysical paradigm where the bat, it's a two-choice experiment where some of the stimuli are stationary, the others are jittering. Here are the percent errors, and these data show that the bat is able to discriminate glint spacings, like in one echo, a spacing in a glint of 1 to 2 microseconds. Again, a very interesting and controversial result. The lab has done neurophysiological experiments to try to probe into the neurophysiological mechanisms of this effect, but I'm not going to talk about that today because I don't have much time. I'm going to point out, move to the most recent work in the lab now. And the most recent work is looking at how the bats perceive the entire sonar scene. Bat sitting on a platform, given 2 echoes and asked to discriminate them, does not tell us what the bat does in its natural environment. And you can see from the schematic that in the natural environment, if the bat wants to get the echo to detect this insect, the bat also has clutter echoes from everything in the surroundings. And for this species, the problem is the bat's beam width is so wide that when the bat makes a call, it's insonifying the entire sonar scene, not just the target, but also the clutter echo. So how does the bat do it? How does the bat do it in a natural environment? And I want to point out one really big advantage of being a Brown or great undergraduate students. And with Jeffrey Knowles is one of our undergraduate students, did a whole senior thesis on bats' ability to form spatial maps in a very complex laboratory environment. Okay, so how do bats distinguish a single target in a complex sonar scene? Here's 2 experiments that I'm highlighting that talked about this. One experiment, and what you do is that the bats are challenged to fly through an environment that's filled with these hanging plastic chains, which give out really intense clutter echoes. And in this particular experiment, the bat wore a telemic on its head and was flying through these chains that produced a lot of clutter echoes. The bat found it easy, they fly fine, not any problem at all. But the data showed one interesting way in which bats can reject clutter, and this is frequency hopping. And frequency hopping is a sonar process that was first described in the 1940s by the actress Hedy Lamarr. And she got no credit for it because she was so beautiful. People figured she was an airhead. And so the male sound engineer who worked with her got most of the credit for it. But they showed that a good way for a sonar system to work was when the sonar system is challenged with clutter, to simply change systematically the frequency of the sonar pulse. And that's what this experiment showed that bats do in these very cluttered environments. They'll frequency hop, alternating the lower frequency of the first harmonic. In another experiment, which is published with Mary Bates, is one of our graduate students. The idea in this experiment was to look at the role of the 2 harmonics, FM1 and FM2, in the bat's calls, and the role of the 2 harmonics in rejecting clutter. And what they showed in this experiment is that changing the delay between the 2 harmonics, which you can do electronically, gives the bat an ability to defocus clutter echoes while still focusing on the echo straight ahead. Okay, so it's 2 ways to distinguish clutter. New work we're doing now is on bats' resistance to temporary threshold shifts. And so these are both psychophysical experiments and flight experiments where the bats are blasted with frequencies and levels of noise which would make a mouse deaf for days. And the bats don't care. Even 2 minutes after being exposed to the noise, the bat's thresholds don't change and the bat's flight behavior doesn't change. And we're very interested in looking at the mechanism of that effect. Finally, Jim has developed a model for sonar processing. It's called the SCAP model. It's based on detection of nulls in spectrograms. We're working on this model a lot, but I'm not. Jim is working on this model a lot. The model at this point is only a monaural model. That's obviously here through 2 ears. And so we need to revise it or expand it to take care of the binaural decision. Another interesting thing is come up with some new work in collaboration with Darlene Catton, which is also going to make us change the peripheral filter bank. Imaging work shows that big brown bats and pipistrelles, Mexican free-tailed bats, have a very unusual ear where the stapes inserts into the cochlea, not at the base of the basilar membrane, but a third of the way up the basal turn. And porpoises do have an ear like that as well. So that raises all sorts of questions about directions of the traveling wave along the bat's basilar membrane and issues about how transduction actually occurs. So now we're going to move towards our 3 pioneers, and here are the titles of their talks. And please welcome Alan Grinnell.
I think we should just let Andrea continue for the next.
Do I have to click on it?
No, I think just wait a couple of seconds.
Just wait, okay,
Control booth somewhere.
okay, he'll do it, okay, great.
Thank you, Andrea. Thank you to the organizers of this wonderful conference in this beautiful venue. I was struck especially by that picture from Frascati of the first International Bat Research Conference, to which I consider this sort of the bookend. I entered Griffin's lab 68 years ago, asking if he wanted an unpaid laboratory assistant. And seeing the progress that has happened in the field since then is just mind-boggling and very, very satisfying. This has been an exciting conference, and I'm really delighted to have been able to participate. Thank you for the invitation. I want to talk today about the importance of FM sweep. Everyone knows that the FM sweep is critical to all sorts of information, but I want to discuss in particular the work of that I think has been pretty much neglected in the field. A cluster of papers done in the mid-1990s by Roald Roverud, who happens to have been a student of mine 10 years before that, following which he did a postdoc with Gerhard Neuweiler and then came back to do independent work, which I think it's interesting. It's way off in left field compared to what other approaches have been. I think it has some interesting implications and depends a lot on how much you trust the results and what the implications are. Now, this is an example to me, an example I like very much, showing the lability, the plasticity and the adaptability of the emissions of bats. This happens to be a black mastiff bat recorded in Belize by Brock Fenton, but it shows the search. This is tricky, isn't it? Shows the search phase.
The lower green.
Yeah, right. The shallow FM quasi CF, which as soon as something is detected, shifts into a steeper FM sweep, then a very large sweep, which eventually transitions into the buzz. Now, the important thing is the information that you get from the FM sweep, and it's generally agreed that this is a variety of information. I'm going to concentrate on target distance determined by echo delay, but of course, binaural comparisons of both intensity and time could provide a great deal of information about the direction in both horizontal and vertical dimensions. And target features appear in echoes by notches and peaks in intensity at different frequencies. And when I say spectral cues here, I'm sure most people realize that this is translated into times of arrival of frequencies in the echo due to the intensity trading relationship. The weaker the sound, the slower, the longer the delay within the nervous system. We start with some basic information, that is that the bats, almost all, well, I don't know any exceptions to the rule that bats develop downward FM sweeps. Some of them also have some upward sweeps, but whether those are actually useful for distance determination, I don't think has been determined. And the white noise pulses also are ineffective in producing information about delay. So it really needs to be the structured FM that bats emit. And as also most of you are sure aware, this, as in the case of the black mastiff bat, the FM sweep can change a great deal too. So the model that has come out of this is that, and I think it's sort of consensus model, is that the emitted sound somehow establishes a template and that the delay is recorded redundantly at all or most of the frequencies within the structure of the echo. And it's been established that Bjorn Siemers and Schnitzler in their beautiful experiments show that target resolution is proportional to the amount of bandwidth in the FM. So it's not only it has to be FM, but the larger the bandwidth, the better. And I'll show some of their data quickly. The Natterai has a very steep, these are 5 species of Myotis, differing in the bandwidth of their FM sounds and their ability to resolve and capture insects in front of a clutter or a wall is proportional to the bandwidth in a linear fashion. So this is the model that I suggested, and I've stolen this slide from Jim. Shows what a parasitic relationship we have in the bat world. But it shows the presumed redundancy in echo delay measurements as a function of frequency in all of the harmonics. And he has gone on, as Andrea implied, to show that if you use artificial stimuli and alter these, that the first harmonic is critical. That is, the fundamental is critical. The second harmonic adds to it, but by itself cannot provide, at least in most bats, would not be able to provide adequate distance information. And in fact, if you gradually chop away the frequencies in the fundamental, you also degrade the performance. So multiple frequencies are definitely necessary. But I want to talk now about, oh, this is again stolen from Jim, the idea that you have an auditory filter bank, each frequency is sampling redundantly with the others and comparing it with the template of the emitted signal. And this has the effect of de-chirping, sort of giving you simultaneous or potentially simultaneous measurement by some integrating element within the nervous system. And concentrating the energy into a specific time, which can then be compared across a bank of delayed tuned neurons. Okay, the question then is how, the question that Roald Rubberud was addressing was how many redundant measurements are necessary? Is there a minimum number? And how can one determine what that is? And in particular, given the fact that the tuning curves of most neurons one studies, at least in the colliculus, are relatively broad, and the delay tuning half width is not all that sharp either. How do bats provide or reduce the accuracy of delay that we see? And believe me, none of us have gotten to the point where we can explain nanosecond resolution, but that's something to keep in mind all the way through this. So this is a series of papers, 1993, 94, 95, if I get to it, one in 1998 or 9, done with a Noctilio albiventris, a neotropical bat, a smaller relative of the fishing bat. Uli and I and Eli Calco have studied this in Costa Rica. But these were done back in the lab and done after he had become independent. So I had no particular input in this. And his approach, his experimental approach, this is Noctilio albiventris. This is actually an insectivorous bat, but it often catches insects that dive when they sense the presence of the bat. They end up on the water surface and it comes along and sweeps them up. Formidable teeth. And this is the kind of emissions that they have. This is a record that we took in Costa Rica, and it shows the typical search phase, which is often either just a quasi CF pulse about 4 milliseconds long, or a CF FM pulse, usually in pairs, sometimes preceded by another CF FM pulse. In the laboratory, and this is just showing with detection how the constant frequency component is shortened a little bit, and then it goes into a complete FM signal. And once the prey is caught, you go back to series of purely CF, and then the combination CF-FM. This, in the search phase, is going about 5 or 10 of these pairs per second. The phase here, after detection, goes up to about 30 per second. What Roald did, which is kind of clever, but quite unlike what anyone else thought to do, was to use absolutely natural behavior, untrained bats with no rewards. It's just a matter of when they're in on a perch in a laboratory situation, if they detect something coming toward them, they often respond by showing by this approach phase response, going from pairs of CFFM pulses to at 5 or 10 per second up to about 30 hertz, 30 responses per second. So the question is, how can you alter, that's with a natural echo. What he was doing was presenting a artificial sound, simulating a bat's CF-FM sound, 4 milliseconds CF, 4 milliseconds FM, and presenting that to the bat when it was emitting sounds as if it was an echo of a target coming toward them at a rate of about 5 meters per second. Between 5 meters distance and 0.3 meters distance. And if this was a sound that the bat interpreted as being an echo of its own sound, or at least close enough to possibly represent something toward it, it would respond by changing its emission to the approach phase behavior. So it's just a natural response to what it perceives as something coming toward it. And Roald tested both the sort of smooth FM portion, but then wanted to test the necessary number of frequencies in the FM component to give this behavior. So it looked like saccades, but this is in fact a step in frequency. So the step size could differ, the step duration could differ, and the total number in the FM, the pseudo FM component of these echoes could differ. In the first experiments, he was varying the number, but keeping the 4 millisecond CF component the same. All the sweeps, in quotes, were the sort of normal range that the bat would emit in captivity, which is from roughly 75 to 55 kilohertz. And the control for this was the pure FM, just the normal FM. So he's essentially reducing the number of frequencies by using steps, skipping some of the frequencies. And on the y-axis here is the percentage success in evoking an approach phase response by the bat. And this is not an all or none phenomenon. The control, normal sort of simulated CFFM was only about 69–70% effective. And one single jump from 75 to 55 was about 30% effective. It's most of the change in effectiveness is between 100, 125. 99 is indistinguishable from the control. 90 is somewhere in between, and then by 80, pure tone steps, you've gotten to the point where it's no longer very effective, or at least not any less effective than really completely abnormal exposure to very few steps. But during this time, the individual steps, because you're varying the number but keeping the time constant, they're varying in duration from 32 microseconds to 200 microseconds. And the step size is varying from 160 hertz to essentially 10 kilohertz. So is there anything magical about the number, or is it just you need these frequencies? In another experiment, again, preceded by the 4 millisecond CF. All of these are going to be preceded by the 4 millisecond CF. But now comparing the response to 99 or 100 tone steps and 80, the frequency, the size of the steps was varied from 100 to 200 to 400 hertz. And in all cases, the 99 steps worked well, the 80 did not. Again, there are differences here in the extent of the sweep. The 80 steps always gives you a little less, but still there's great overlap between the 80 and 99 step. So it looks as if it's really the requirement that there be 99 separate frequency steps, and 80 is not adequate. Individual step duration, again, you can vary it over a considerable range, and it still does much better with 99 than with 80 steps. Duration of the sweep is shown as here. Frequency difference between steps, looking a little bit more detailed. 50 hertz is not adequate. This, in this case, gives you just a 5 kilohertz step, 5 kilohertz sweep. 100 going from 75 to 65 is perfectly adequate. 75 to 35 with 400 steps, also perfectly adequate. Conclusion here is that 50 hertz appears to be too small a step to count as a separate frequency for purposes of redundancy of measurement. And because the frequency sweep is so small, is that the explanation, or is it the number of steps? And when he altered it, so he combined 50 hertz with in every other one being 350, 300, 200, 100. These combinations of step lengths, again, the ones which included 50 hertz were inadequate, or were less effective. So the conclusion was that a step of only 50 hertz is too small to serve as an independent step, meeting the requirement of more than 90 steps to simulate a true FM sweep. In another paper where he presented the data differently, he again tested the duration of the frequency step in noctilio, and found that durations between actually between about 40 microseconds and 80 microseconds are fine, but when the duration got as long as 90 or 100 microseconds, the sweep correspondingly longer, that did not work. So it's telling us something about the interpretation of frequencies coming back in the sweep. I guess the band is arriving. Wait to see that dancing. Again, I can go rather quickly through these. The point is that he varied duration in a variety of ways, checking the total durations. And again, the 90 and 100 microsecond durations were too long in the case of noctilio. 38 steps, going over 10 kilohertz or over 40 kilohertz with different size steps. The durations was just an absolute requirement that it be less than 80 microseconds. So the general conclusions of these experiments with noctilio are shown here. There appears to be a minimum requirement for the number of individual frequency bands. And these can be as close as 100 hertz from one another, but not 50 hertz. There's a requirement that the duration be less than 80 microseconds or less. And the conclusion is the neuronal FM pattern recognition requires integrated activity of 99, or at least to some extent 90 or more of these filters, but they can be distributed and are distributed over a wide frequency range. You can have them all within 10 kilohertz from 75 to 65, or 75 to 35, and it still suffices. Okay, then for comparison, we did similar experiments with Eptesicus. As you can see in this case, the performance wasn't any worse at 90 to 99 than it was at controls, a little better, if anything. But notice that the abscissa is completely nonlinear. Between 85 and 80, you get a loss of this ability to interpret the sound as being a natural echo. And in Eptesicus, the frequency difference between steps requires that the difference be at least 450 hertz. In noctilio, it was 100 hertz. Duration could be up to 50 microseconds, but above that, it didn't work nearly as well. So there are clear differences between Eptesicus and noctilio. These are the only two that he studied under a variety of situations. 16 greater than 50 microseconds did not work. So the overall conclusions were that Eptesicus needed somewhat fewer steps, fewer frequency filters than noctilio, but the minimum step size was much larger. They have steeper, larger sweeps than noctilio, so that makes some sense. The minimum step duration is different, and the FM sweep rates overlap, which in noctilio went from 2.5 kilohertz per milliseconds on, and in Eptesicus it had to be greater than 9 kilohertz. So how do you get such sharp filters? That's a good question for the field as a whole. But one of the characteristics of units in the inferior colliculus, this is from Tederina and work of Bodenhaber and Pollock, is that the units in response to an FM sweep, if the curves here are the tuning curve for a unit, with a downward FM sweep, the response comes as a single spike when the sound crosses the high frequency order of the unit. It's okay. I will do this very quickly. The result of this is that the tuning curve, the effective tuning curve, is much sharper, and in fact, not all that much less steep than the tuning curve from the CF component of Terra Nodus. similarly, work by Ellen Covey with Huffman showed very sharp tuning to FM, small FM oscillations around the high frequency component. And an independent way of doing this is to measure the minimum difference limit for the frequencies in different species. Van Stebbet and Schmidt, the 2001 paper, found the difference limit in my Aptascus to be 118 hertz, pretty close to what Rold and Rabatoy had found. Bodenheimer and Pollock found the frequency difference limit in Cheddarita to be around 900 hertz. And of course, the CF bats in their constant frequency region have accuracy of 100 hertz or less. The delay tuning is also, if you look simply at the delay tuning of a single neuron, it's steep, but still there's a considerable 50, at the 50% point, it can be 2 or 3 kilohertz, even in a sharply tuned one. Family of them, they come at different delays as one would like. And this was interpreted by the authors as being evidence for a lack of tonic or delay organization in Aptascus. But in fact, I think it's evidence of redundancy of this. So you do get a spatial organization of different delay records, which would make it much easier to sort of find the point, the distance corresponding to the activity of a family of neurons. So I will stop at that point, although if discussion permits it, I would cite one more result from Roald, which shows that you can get this activity, approach activity noctilial with upward stepping frequency.
They're gonna change it themselves, right? They'll do it, okay. I don't have to do it. Yes, okay, and I'll now introduce Jim Simmons, talking about where's the target in the back.
Locking the door, that's a new approach, right? All right, ready? All right. Okay. All right, there's the title and the stuff, okay? So I don't like to talk about myself, but I'll give you a few sentences, all right? I'm an experimental psychologist, trained in psychology in Glenn Weaver's lab at Princeton University. And the initial, the beginning of a lot of the work I'm going to talk about is entirely from psychology. In my life, I have never taken a biology course, and you need to remember that. In other words, if I say something that sounds like I'm not talking clear about biology, there's a reason. Okay. Let's find out what we point this. Okay, there we go. Okay, here's the question. Okay, how is the echolocating bat's perception of echo delay represented in neural activity? And there's a clue from Bob Kopranica's remark back in 1981 at a neuroethology meeting. The vertebrate nervous system is not critically organized at the single cell level. All right, there's an added reason why this is important here. And it's that in the bat, in big brown bats, when you present the bat with FM pulses and echoes, the vast majority of neurons produce only one spike. And those spikes are distributed according to the latency of the cell, according to the width and delay of the FM sweep, and according to the delay of the echo itself. In other words, you have these spikes occurring all over the place. If you have a target that's at an echo delay of 10 milliseconds, you're going to have combinations of pulse and echo responses stretched over 30 or 40 milliseconds. And it's immediately clear that if you expect to solve the problem by figuring out which cells are responding and not when they're responding, you won't succeed. All right, so the initial work on the problem of where in the vertebrate nervous system, this is the rat, the distribution of remembered perceptual information was first really well studied by Carl Lashley, who was at Harvard University for part of the work. He developed a test where bats sit on a platform, rats sit on a platform and jump to one of two visual stimuli. If they choose the correct one, the platform is there. If they choose the incorrect one, there's no platform and the rat falls to the ground. Now, rats are not terribly disturbed by this because they do multiple tries of the experiment. So it's not like, bzzz. It's more like, well, whoops, we'll try again next. Okay, anyway. Lashley's main conclusion is that the representation of the information in a remembered perception, these are memory experiments, so you can't really separate what is perceived from what is remembered. But he clearly demonstrated that the representation is essentially volumetric. Your ability to impair the perception and the memory is pretty much dependent upon how much of cortex is taken out, and to a large extent, not which cortex is taken out. And this has been variously called equipotentiality or mass action. This is work done in the 1940s, 50s, and 50s, and it is the beginning of what in psychology was called the search for the engram. The engram being the memory. Now, no biologist would put anything like that. Where's the engram? That's not the way it works in biology, but that's the way psychologists thought about it. Lashley had one extremely important graduate student, Donald Hebb. Now, Hebb basically wrote one of the first clear English language, non-mathematical descriptions of how the brain might do this. And he talked about cell assemblies and phase sequences and things of this kind. It's a great book. I read it as an undergraduate, and it's what, in my second year as an undergraduate, second try, and it's what got me started on this stuff, okay? All right. In the late 1950s, Letvin, Maturana, Hubel, McCulloch, and Pitts published a paper called What the Frog's Eye Tells the Frog's Brain. And this, I think, can fairly be described as the first published event of what we think of as modern neuroscience. Essentially, the visual, that the frog's eye decomposes the visual image into visual information into features of edges, contour, motion, and so forth, and not as a picture. There's a transformation by features. All right. This was picked up by Hubel and Weizmann, who are famous for having launched this giant, I wouldn't say cottage industry, I would say large-scale industrial effort to figure out how the visual system works. And we all know about that story, about how feature analysis is an important aspect of thinking about vision. And as an aside here, Hubel and Weizmann were at the Walter Reed Army Medical Center working under the supervision of Robert Galambos when they start their single unit work. That's important to remember. They did this because the alternative was to go to a MASH unit in Korea. Now, the feature approach, I think a good way to describe it is the feature approach is well represented in the work of Nobuo Suga, who was a postdoc of Don Griffin's contemporary with Alan. And he basically developed a really nice feature map for the mustache bat's auditory cortex, velocity, angle, Doppler shift, range, and so forth. And Suga will often show up, this map, we'll come to it in a minute, pictures of the map, show the map, say, this is like the cockpit display for a pilot in an airplane. And then he would say, where is the pilot? Which I think casts rather well what our problem is. Okay, now the problem with the feature approach was first The weakness was first discovered or realized by Christoph von der Malsburg, and then realized in physiological experiments by Wolf Singer. Basic problem is if you have all these features being stimulated, those same features are being stimulated by lots of other things in the visual field too. And the problem is the composition of your obvious perception of the object in which the features in here. The only way that would work is if the neurons are responding synchronously for the features of that object. All right. I won't go into details about how that's gone, partly because I don't understand a lot of it. But the main point is that we're talking about a system where the time of occurrence of nerve spikes, at the very least, must define what the object is and probably determine what its features are when they are perceived. All right. So to test whether the timing of neural responses acts as the vehicle for composing percepts, we need to measure the acuity of perception of time itself. And then we need to figure out what happens if you perturb the time in different feature channels. We use the auditory system. Now, I didn't do this when I was a graduate student. I didn't have a clue about this stuff, all right? I probably couldn't have said any of this 15 years ago, okay? But in the Bat's auditory system, we can change the time of occurrence of nerve spikes in different frequency channels relative to others and assess whether how that affects perception of delay. And that's what we're doing here. All right. So the early experiment, you've already seen this first, the bats on the Lashley jumping stand can detect differences in target range in the range of a centimeter, centimeter and a half. And if you replace physical objects at different distances with virtual objects, you find that they can detect changes in delay in the range of 50 to 80 microseconds. All right. So far, so good. Okay. By the way, this was the first use of virtual methods in hearing, but I'm not claiming anything. I'm just saying it. Okay, just saying. All right. All right. Now, back in the mid 1970s, what did I do in 1977? Okay, in the mid 1970s, George Pollack was doing work on the neurophysiology of the inferior colliculus in free-tailed bats, Tidarida brasiliensis, and he never got trapped into the idea that you perceive echo delay from delayed tuned cells, partly because they hadn't been discovered yet, and partly because it didn't seem like a plausible approach that would work very quickly. So basically, he presented bats with artificial pursuit sequences for free-tailed bats, and then measured the latency jitter of the spikes in the inferior colliculus. And he got average jitters that would give you a delay value, a delay acuity of about 10.9 centimeters at one particular distance, and then 8, 4, and 3. So basically, you're talking about individual neurons being able to do 4, 5, 6, 7, 8 centimeters of delay acuity at different distances. And if you think, well, there must be lots of other neurons, it's pretty clear that this finding that the animals could do a centimeter or two in a stationary psychophysical task compared to what individual neurons could do, this is great, okay? If we had stopped here, my life would have been vastly simplified, okay? But we didn't, okay? We used electronic delay lines for delaying the echoes in the virtual echo experiments here. And the delay lines had settings for 100 microseconds per step, 10 microseconds per step, 1 microseconds per step, and a continuous 0 to 1 microsecond final stage. We never messed with that because we had no way of determining whether a 0 to 1 microsecond change produced a 0 to 1 microsecond change. But when I moved to Washington University in St. Louis, we had developed a means for measuring echo delay or signal delay accurately and using the phase of constant frequency signals, that we could actually change the delay of echoes going to the bat in steps trustworthily of a microsecond or two. And we pretty quickly found that big brown bats can detect changes in echo delay of the order of a microsecond or two. And if you plot the performance of the bat in determining delay values around a 0 value, this is about 3 milliseconds of total delay, you're talking about jitters of 10 microseconds, 20 microseconds, 30 microseconds, and so forth. The plot looks like this if the two echoes have the same phase. And it looks like this if the two jittering echoes are also jittering in phase, all right? Which means they can perceive changes in phase. All right, that sort of resulted was experiments that Cindy and Uli Schnitzler did in Tübingen. And I'm sorry that Jim Finnern is not here, because he would tell you that the bottlenose dolphin produces exactly the same results, just shifted to higher frequencies and with better resolution against the side lobes, because their signals are higher in frequency and broader in bandwidth. In other words, whether you like it or not, these animals actually do perceive the phase of echoes. Let's go back through this again here, just one more time like that. I won't go into the details of this, but we did lots of things like restrict it to a frequency range of only around 55 kilohertz, and you actually get a narrower band, longer range of phase cycles. So I won't go into the details of that. All right, so here, when the crucial feature here, the question is the time of occurrence of nerve spikes in the bat's auditory system conveying the information into the bat, which ultimately gets perceived. I'm not saying anything about how it's perceived, but which ultimately gets perceived. By changing the amplitude of one of the two jittering echoes, you can shift the perceived delay. We had measured amplitude latency trading in behavioral tests, and it's about 15, 16, 17 microseconds per decibel of echo reduction under these conditions. And what happens here is here's the phase. We all have the same problem with the thing, don't we? This is, all green, but normal. All right, okay. Boy, either place or color memory is bad. I don't know which. This is the jitter experiment plotted as a compound curve on top of the green cross correlation function of the echoes. The whole thing shifts over the green function and the red function compared to this by the amount of 17 microseconds per decibel, which is about the best you can do to show that it's carried into the nervous system by the timing of nerve spikes. All right, and now, at one point, once I got to Brown University, we decided we fool around with those right two dials on the delay lines. And we were, frankly, initially somewhat disappointed to discover that we could get changes in echo jitter to be detected. Now, I want to be clear what I'm saying here. They're detecting changes in jittering echo. I'm not saying they're detecting an increment in delay of 10 or 20 nanoseconds, only that the change in delay is detected relative to no change. And what we find here is this rather complicated plot. Let's go up here, look at it here. Here we have signal noise. Here we have signal noise. Here we have signal noise ratio here in decibels. This is true signal noise ratio, T over 2 over n 0, 2 pi in the denominator. That pesky 2 pi was the main reason I had to do physics and do freshman year twice. I did not like the idea that in physics you could take in or take out a 2 pi and everything would be so different. Pi, after all, is a number, right? Difficult number to specify exactly, but it's a number. All right. So here you have signal noise ratio here. You have a bunch of curves here which show the performance of different models. This is the Heidi Hackbarth filter bank model with different up here performance in echo delay acuity, assuming that the signal noise ratio and bandwidth are all you're correct using. It's called the Kramer-Rao prediction. And then the SCAT model, the SCAT the bandpass filter model, which has, in this case, we use 100 channels because that's what Roald Robert's numbers suggested that test is probably used. So you get the signal noise rate, you're getting the bandwidth increasing like this. This graph here, this dashed blue line is the prediction for a coherent cross-correlation receiver. Slightly above that is the prediction for a semi-coherent cross-correlation receiver. You use the term semi-coherent here because the cross-correlation function is determined before the phase is destroyed. It's not like you destroy the phase and then you try to do the cross-correlation. Well, but what we found in these experiments is that big brown bats could detect jitter and noise at this point here and at this point here, which approximated that of a cross-correlation receiver. Now, give or take a few dB. You know, this is
These are animals. If you think that you're going to get a 2 dB change making a big difference in performance, it's probably not the right way to think about it. But what it does show is that the animals behave as though they, and also a filter bank model, approximate cross correlation, provided the filter bank individual frequency channels have low pass smoothing no lower than about 10 kilohertz. Any questions? Just it's a baby in the audience, you know. Then up here, very low signal to noise ratio, performance deteriorates because you leave the Kramer-Rao region and enter the side lobe region. It's a long story. Okay. Now, when we at Washington and at Brown University, we had a graduate student named Mark Sanders, and he was working on perception of echo phase changes and recording local field potentials from, I think, best described as inferior colliculus. We specify as sort of the cochlear nucleus, but it really is probably some brainstem mid-brain location. So here we have a stack of these local field potentials with.
Can you move down so that Uli can listen?
Yeah,
'Cause
okay,
we
I'm
can
sorry,
only
yeah,
hear
okay,
when
yeah.
it's on the-
All right, how's this? Okay, let's go back to here. All right, here we are, right here. These are these local field potentials. Okay, that's better. Local field potentials for tone bursts changing in phase between 0 and you can see the phase changes over here like this. And these are the evoked potentials here, 12, 14, 16, 20, and 30 kilohertz. And what you notice here, that the responses to the tone bursts at 12 and 14 kilohertz are demonstrably changing in phase, in the sense that the amplitude of waves of the evoked potential is phase sensitive. All right. All right. If you plot the integrated energy in the evoked potential in this time range, a few milliseconds after the stimulus, compared phase condition reversal compared to the control condition, which is no phase shifts, you find that that this is an estimate of the low pass filter characteristics of the band pass filters in the BAT, which we use for the cochlear model, around 15 or 16 kilohertz. In other words, all you need to do is have low pass filtering up 10, 12 kilohertz, and you achieve with these FM sweeps the same performance you would get if you had an ideal receiver. All right, so that's 3 of the 5 fatal objections to the results of the jitter experiment, because most of them had to do with, well, you can't do it with low pass filters around a kilohertz because that's what humans have, right? And bats must have that too. Well, no, bats have a roll out a little bit 10 times higher. All right. Another thing that turns out to be interesting is that Mark Sanderson found is that the phase effect was in the offset response, not in the onset response to the tone bursts. So when you change the duration of the tone burst, you see here the phase sensitivity follows the offset and not to the onset. This is important because Paul Four and Ellen Covey and many people, not many, many people that tell me, several people have studied duration tuning in the inferior colliculus and found the surprising effect that many neurons in the inferior colliculus are tuned to different tone burst durations. And in a bat, this might seem like not too useful because the last thing it needs to do is to estimate the duration of its calls from the inferior collicular response that's already been determined. So, and Paul Four and Fierslav have pointed out that that might mean that this kind of response, this offset response, is coupled to determining echo delay, and not specifically to the duration of a tone burst. So here we find sensitivity of phase to the offset in a response to a tone burst at frequencies up to about 15 kilohertz. All right. So the plausibility, in other words, you can't say it's auditorily impossible anymore. It may be wrong, but it's not impossible. All right. The next question concerns what happens to auditory, to delay acuity. This is a graph here, which shows echo delay acuity or hyperacuity in the jitter experiment in nanoseconds, in the range of 20 to 50 nanoseconds, as a function of the reduction in bandwidth caused by either low pass or high pass filtering. And what this plot shows is that if you only remove
high frequencies, FM2 frequencies, performance goes down at this slope. If you remove FM1 frequencies, this would be the slope you would expect here, but instead you get 3 times that. Because when you remove a frequency from the first harmonic, you also remove the frequency in the second harmonic from the point of view of the bat. As Cindy and Uli showed in their experiments, the second harmonic is incapable of, basically, if there's no first harmonic, the bat doesn't think it's an echo at all. They just won't perform. Let's deal with that. Okay, so the final step here is this. The second to the final step here is this. Can we now reconstruct the performance of the bat, which is the 2 0-degree phase and 180-degree phase jitter results, behavior results, using only the spike times from the peripheral auditory filter bank? Yes, if you plot the detection of 0, which is here, it's easy, there's no data. If you show phase shift changes in the SCAT model as estimates of delay based upon changes in phase, you get approximately the prediction you expect for the behavioral results. So a model auditory system can perform the same as the bat. Now, the obvious problem here is what went wrong, okay? As several people in this room have pointed out as recently as yesterday, how can you do this with nerve spikes where the duration of the spike is 0.5 millisecond and a good rise time is 100 microseconds? You can't. All right, I'm not prepared to say that 10 million neurons of the Nelson-Kang type switched up to high frequencies will explain this. What we do instead find is that when you present stimuli to the inferior colliculus, you get 1 spike per cell, but if you pool the responses across cells, you get an oscillatory pattern. Now, I'll come to what that means at the single unit level in just a minute, but what we see here is measurements of oscillation responses for the multi-unit response in both Corolla and Eptesicus, obtained with a slightly unusual electrode. It wasn't intended for that, but that's what we find. You present a noise burst or a tone burst or an FM sweep to Corolla and Eptesicus, and you get sustained oscillations at frequencies between about 400 and about 750 hertz. These are analog signals that are happening in the bat. They've also been observed in experiments in Cindy's lab by Jin Hong. And once you realize that they're there, you go and look for them, you see them elsewhere. This is a local field potential, which is essentially the multi-unit response. How many units, I don't know. Not 1, not 2, all right. Well, anyway. Here we go. So this is now, we'll go back to data collected in the 1990s. This is work by Mark Sanderson, by Mark Sanderson and by Mike Ferragamo and by Tim Herstein. These are plots here. These are latencies of responses here. This is best delay for delay tuned neurons. Delay tuned neurons have their tune delay value in a triangular space of latency and delay that's driven by neurons in the inferior colliculus. Neurons in the inferior colliculus themselves have latencies from about 4.5 or 5 milliseconds to 25 or 30, but there's a large number of neurons here. And when you look at the plot of essentially the PST histogram for the on response, each of the points in here is a single cell with a single latency. We're not talking about cells oscillating. We're talking about a pattern of oscillations collected over a number of different bats during the course of a year. So it's very robust. You get these oscillatory patterns here. When you subject this red curve here, almost done, okay, to a Kepstrom analysis, you see that you're talking about time intervals between 2.7, about 3.2 milliseconds. Those are the periods for the oscillations that showed up in the collicular responses of Eptesicus and Corolla. The final thing is, this is where we think the target is in the bat. All right. You get a response in the inferior colliculus, which is projected to the medial geniculate body, about which we know almost nothing in bats, and then to the auditory cortex. And in the auditory cortex, it produces a spike. Neurons in the auditory cortex project directly down excitatorily to the inferior colliculus to the corresponding frequency location. So it's an excitatory reentrant map in the sense that a lot of people have talked about over the last 30 years or so. In the meantime, that same location projects to the ground, literally the outer layer of the inferior colliculus as an excitatory signal, which then projects an inhibitory signal to the same location as the just previously arrived excitatory signal. And this thing goes back and forth several times. All right. So when a bat emits a sound, it produces an oscillatory pattern. When it gets an echo, it produces an oscillatory pattern. When you get another echo, it produces the oscillatory pattern. The object is in these oscillations. It never shows up at the level of precision we need to consider for bats in anything other than the oscillations themselves. So good luck with finding it. I am retiring in the next year or two, and I welcome you to work on the problem. Thank you. Over here.
Now, please welcome Uli Schnitzler.
I have 10 minutes, therefore I will shorten my talk. I planned to begin with a short review on Doppler shift compensation in horseshoe bats, but Annette has already introduced it, therefore I can go on. And now horseshoe bats belong to the guild of narrow-space flutter detection foragers, and these foragers produce signals consisting of a long component of constant frequency, which is followed by a terminal FM sweep, and the constant frequency part senses Doppler shifts, and the idea is you already have heard about Doppler shift compensation. The resting bats emit the CF part at the so-called resting frequency. And if they start, they lower the emission frequency and keep the echo frequency constant at the reference frequency. If we look at the audiograms, we see at the reference frequency a very low auditory threshold. And that reflects the so-called auditory fovea, an area on the basilar membrane where the frequency representation is very high. And that leads to many highly tuned neurons in the foveal areas of the brain. And the idea is that these highly tuned neurons are very sensitive to the modulations which are caused by the fluttering wings of insects. And if we insanify insects, we can see that the amplitude of the returning echo is modulated, and especially conspicuous are strong amplitude peaks, the glints. At every wing beat, the wing is perpendicular for one instant, and in this instant, the wing reflects high intensity. And the moving wing also causes, in this situation, a frequency change in spectral glint. And the spectral glints depend on the angular orientation of the insect. If an insect is flying towards the bat, the glints go downward, for the wing is moving away from the bat. And it's the opposite. If the insect flies away from the bat, then the glints go upward, and we can, I don't know how I activate my loudspeaker. means. And that's the information which is available for the bat. When I did these experiments, I always had the dream to sit on the back of a bat and fly through the night and get the experience how it is to detect fluttering insects with long signals of constant frequency. And I never imagined that sometimes it would be possible. I got some data from Yossi. These are tech recordings from Bulgaria. And we see here the flight tracks of 3 individuals of bats. And they had an attack on the back, which recorded the echolocation signals, but also the GPS data of their flight. And if you go sit on the back of the bat, you get a good impression what's going on when a bat is foraging for fluttering insects. According to their motor and echolocation behavior, we discriminate several behavioral situations. When the bats fly out, they often perch at vegetation, scan around, and look for fluttering insects, go and catch a fluttering insect. And then the next step is they fly on and make a commuting flight. And the commuting flight is always along either a horizontal or vertical structure. A different way of flying is over water. For that, the echoes get reflected away from the bat. They don't get the echoes back. And in a very, very rare situations, I think, it was only one bat which left the ground for a few seconds. You have a situation where the bat is flying in the open and gets no echo back. And you can now look how the echolocation behavior of bats is in these different situations. Before I want to show you the flight behavior. For instance, this bat was flying along vegetation, commuting flight. Here it's perching. For every point is 15 seconds, then it's going on with the commuting flight, perching, commuting flight, along vegetation, and here along a field. And finally, the bat comes to its main foraging area, which is a high voltage power line over here. And at the poles, there was some vegetation, and in this vegetation, they perched and went for insects. The other bat flies here along a vegetation line near the Danube River over some fields, crosses the Danube River, and is on the other side in a small forest. Also, that's the main foraging area. The bat was flying up to 40 kilometers to get to this foraging area. Then it crosses, the bat crosses back over the Danube River. And that was one of the rare situations where the bat crossed a steep slope and was out in the fresh, in the open air for about 40 or 50 seconds. And now we look at the data. These are the sound recordings of a perching bat. The bat emits a signal. No. The bat emits signals with a resting frequency of 79.5 kilocycles. Pulse duration is between 45 and 55 milliseconds. Pulse interval about 100 milliseconds. And you see one pulse after another. And the interesting part is the tech was so sensitive that we do not only see the emitted pulse, but also the echoes behind it. And this perching bats got echoes up to a distance of about
4 meters. In one situation, that was the bat which had its main foraging area near the high voltage power line, I found a completely different perching behavior. Instead of making signals around 50 milliseconds, this bat made very, very long signals. And these signals had no echoes. So I assumed that the bat was hanging and looking out into the open. Unfortunately, I couldn't find any insect catches, but there seem to be different types of perching. Another typical situation for perching is scanning behavior. Here the amplitude bat looks to the left side, poor reception of the microphone on the back, then it comes back and to the right side, and each cycle lasts about 2 to 2.5 seconds. So that means the bat makes about 4 or 5 perching movement, scanning movements per second. When the bat starts from the perch here, they lower the emission frequency and you get echoes back either from the ground, if they fly over the ground, or from the vertical contour line of vegetation. And this echo is about at the same height as the emitted frequency. Here we have single pulses with the echoes from the background. And here are pulses in flight, and you see the lower emission frequency, and here the Doppler shifted echoes frequencies which are cut. Here there's always the highest maximum frequency, and this highest maximum frequency is about the same as the rest. But that's it, you know. Sorry? Listen there. Oh, now we have a sound sequence of a bat flying over a field. Again, we have the emission frequency, which is lowered in order to compensate for the Doppler shifts, and the echo frequency is in the range of the resting frequency. The idea of Doppler shift compensation is that the bats compensate the Doppler shifts from targets ahead. I can measure the flight speed of the bat and can calculate this Doppler shift. But the Doppler shift which I measured was slightly smaller. It was about 9% smaller. And by calculating the relation between the maximum frequency and the Doppler shifts from ahead, I can calculate the angle from which the echoes come back. And this angle is at about 25 degrees if the bat flies over a horizontal field. And I can also measure the flight height with the sinus alpha. And this bat was flying at about 1.57 meter. The problem with these calculations is I'm not sure how precise the speed measurement is. For the speed jitter, so the GPS data are not very precise. Here we have the situation of a bat flying along a vertical contour. There is one thing which you should look at. I not only get the echo, the direct emission frequency, there is always also an echo from the wing. And if you look here, the wing echo is always in the beginning of the signal. That means the signals are emitted with a high correlation with the wind beat. So probably the wind is moving upwards. So the tag gets the echo from the head. And again, also the reflection from the wing. Here another situation, flight along the contour, the same situation. When the bats fly over water, they only get a belly echo back, for all other signals are reflected away. And in this situation, the bats emit 70.8.6 kilohertz. That's not the resting frequency, it's somewhere below the resting frequency. The same situation I don't know why. Sorry? In one situation, I got or 3 or 4 pulses, I got also the belly echo, which is not Doppler shifted, for it comes from 90 degrees. And the belly echo was 6.8 millisecond delay, which tells you that the flight height of the bat was about 1.16 meter. All the flight heights we measure correspond well with observation which we have from bats in Switzerland. There we observed bats flying along a vertical contour, and they had a flight height of about 1 to 1.5 meter, and a distance to the vegetation also about 1 to 1.5 meters. So these data confirm our observation from Switzerland. And here we have the situation where the bat had no contact to the ground. And again, it's very similar to the situation over water. The bat adjusts to 78.7 kilohertz. So that seems to be just the type of reaction if the bat has no echo. Here again, a bat which starts, you see the signals of the perching bat, then it goes down, and it needs about 6 to 7 pulses till they are down to the emission frequency, where the maximal echo frequency is kept at about the resting frequency. And here we have a landing bat, single pulses, groups of 2 to 3 pulses, and finally a group with many short pulses with a duration of about 10 to 12 milliseconds. Here again, you see the emitted pulse, some echoes in between, and up here, like a sharp cut, the highest maximum, the maximum echo frequency. I don't know why this doesn't. Sorry? Oh, okay. Here we have a situation where a bat catches an insect on the wing. You see the bat coming on, you have the background information, and then it detects the insect and goes up here, catches here the insect and flies away. Here another catch. The bat slows down, catches the insect and flies away. The most interesting part is here we see the glints. These are the reflections which are produced by the moving wing. Every time, every wing beat cycle, the wing is perpendicular to the impinging sound wave and makes the strong spectral glint here. And we see here a glint pattern. The distance between glint is about 20 milliseconds, which corresponds to a wing beat rate of about 50 hertz. So that's what the bats want to hear. They want to perceive glints, and the lowering of the emission frequency has the result that the glints are in this situation, above here again. And here you see it more in detail. Here the emission frequency goes up and the glints come down for the bat narrows in on the insect. The same situation over here. If you look at the data, you have the problem that the bats often try to land at a twig. And then they do not manage it. So you get the feeling, oh, it's a wonderful insect buzz, an insect capture. But if you compare captures and landings, you see a difference. A landing is always begins with groups of 2 or 3 signals and has a long terminal group with many short signals. Whereas an insect catch normally comes from single pulses, and there is only one terminal group, and within the group, the signal duration is reduced. So this pattern helps you to discriminate between landing buzzes and buzzes which are produced when the bat catches an insect. So what did we learn? We found that that's now the result from one bat. This bat has a resting frequency of 79.46 kilohertz. When you're commuting above horizontal or vertical structures, it adjusts an echo frequency which is exactly at the resting frequency. So the bats want to hear the echoes at the resting frequency. They fly over water, the frequency is somewhere in between. I cannot explain why, but that's the result. It's the same when the bats fly in the open. And we assume that the bats adjust the echo frequency exactly in the foveal range where they have many neurons which are highly tuned. And this foveal range is here depicted by the density of such neurons. That's a logarithmic scale. So up here, there are 200 times more neurons than down here. And here it's even 500 times more. And that means this is the area where the bat has many, many neurons which are specialized to detect fluttering targets. And now we have the situation of a resting bat. We already published this figure years ago. And you see here an insect which passes tangentially and has no additional Doppler shift, produces glints to both sides. An insect which narrows in and has a relative high approach speed. In this situation, it's an approach speed of 6 meters per second. The glints go downward and stay also inside. And if the insect flies away, it's just the same. So you have the bat always adjusts if it emits a resting frequency, it has the highest chances to catch the glints which are produced by fluttering insects, either moving towards the bat or away from the bat. And now we look, how is it when the bat is on the wing? Wo muss ich draufdrücken? Okay. And now we have the situation that's new. How is it in bats doing area catches? And we know bats doing area catches also adjust the echo frequency exactly at the resting frequency. They want to hear the carrier frequency of the insect echoes near this resting frequency. And if we look here, we see the glands are already also arranged in the area where the bat has the most sensor for fluttering target. So Doppler shift compensation has a biological significance in resting bats and in bats catching insects on the wings. The carrier frequency is adjusted in the range where the ecosystem or the analysis system of the bats is optimized for the detection of fluttering prey. Okay, very short, a little bit information on the dynamics of this Doppler compensation system. If you play back artificially shifted Doppler signals, one can see that the bats adjust for this positive shift by lowering the emission frequency, and this system is rather slow. For instance, if the bat makes a jump over 3 kilohertz, it needs about 10 signals till the bat is down, and on the way back, it even needs more signals. So Doppler shift compensation going down and going up is asymmetrical, and both systems are very slow. Going back is even slower. The range over which bats are able to compensate for Doppler shifts is very high. That's the world champion of Doppler shift compensation. It compensated for 8 kilohertz. That was the bat Alan and me used to measure the directionality of sound emission. Only positive Doppler shifts are compensated for. Negative Doppler shifts are not compensated. And if you give sinusoidal Doppler shifts, you can also demonstrate that the Doppler shift compensation is very slow, which has an advantage if there are fast changing Doppler shifts. The bat stays. Yeah, the bat stays on the on echo F max on its frequency. It doesn't react to all changes. It's some kind of a low pass filter for flying along contours, and that's very important. So we think that the slowness of Doppler shift compensation is an advantage for the bat. And but it may be a disadvantage for the bat, which starts from a perch. For going down with high speed, it would need a long time till it has the wanted echo frequency. And I looked at data from Arjan and other people, and I found a real interesting result. For if you look, here we see the reaction of a bat without any auditory feedback. And the interesting result of this paper is that bats without auditory feedback also lower the emission frequency. And then I marked the range in which the bat has a positive acceleration on the swing. So I'm absolutely convinced that this proves that the vestibular system has also an influence on Doppler shift compensation, and that has the advantage that starting bats can lower the frequency faster as if they would do it alone by auditory feedback. The Doppler shift compensation, Yossi discussed this and said we have to make a model. Then I looked into literature and found that Doppler shift compensation has a lot of weighting functions. So it will be very difficult to get all this weighting function to produce a model. That's the literature. And we can see that the back experiments indicate that the Doppler shift is weighted according to the auditory input, sound duration, delay, all kinds, binaural influences, noise, and so on. So you have to know all this weighting function to simulate the Doppler shift compensation. And the idea is that the incoming information is weighted in the auditory system, and then it's compared with the set point conditions in the vocal control system. And as long as you have a deviation between incoming information and set point condition, the emitted frequency is adjusted. And the weighting occurs according to the input of the four-wheel system. The four-wheel system is not frequency stable. For if you change the body temperature, the resonance frequency changes. So it's a place-specific system and not a frequency-specific system. And maybe the last sentence, anatomical and neurophysiological data suggest that the faster lowering of FM, the going down, is an active process. Every time some kind of guardian neurons are activated, the frequency is pushed down. And if there is no lowering effect, it goes slowly, passively back, like by a rubber band, then it's pushed again. And this means it's an active process going down and a passive process going up. That's it.
Okay, so we now would like to ask you to come forward, Uli and Jim, and then we can we make it short because dinner is is waiting. So,
are there any questions from the auditorium? Yes, dear. You have to press the button.
So I've got a question about the last talk. So I've always I've also always wondered if one of these CF bats flies through space, what the variability is, or the temporal variability in the Doppler shift it experiences, and this is probably the first. Okay, this is confrontational. This is probably the first time you can actually see what the bat experiences as it flies to natural habitats. So I was wondering whether you can comment on how fast the returning echoes change in Doppler shift.
Wenn das Echo zurückkommt.
The lowering is rather slow, but that's supported by the vestibularis if they take off. But if they are down, it's rather stable. So they don't, if there is a change in Doppler shift, you can see this rather often, that there is a bigger Doppler shift. They don't care. If they go slowly back and are pushed down a little bit faster, but the low pass filtering of the dynamics is very important. I have a hearing problem, okay. So Annette will translate.
Okay, just to see whether I understand. So you're saying that you do see the Doppler shifted echoes varying very quickly in frequency, but the bat is just very slow and does it's kind of okay.
Schnell, aber das ist schnell ändert, aber die Fledermaus zieht nur langsam nach. Oh, okay, good.
You
have to switch on, yeah.
Okay,
and then
so
go
difficult.
Right in front of the microphone.
Maybe I'll just come down there. Okay, so just to make sure I understand, right? So you're saying that the echoes, the Doppler shifted echoes received by the bat do change very quickly in frequency, but the bat
due to the echo situation.
Yeah. Okay. Yeah. All right.
So a question to all. So if this downgoing sweep and frequency is active, could it be a kind of a scanning motion to look for the profile of Doppler shifts, telling you the velocities in the environment, etc.? Could the purpose of the downward sweep of frequency be scanning of the spectral domain in order to get the velocities?
Sorry,
around
I
it?
didn't get the question.
It's question to anybody?
Just
Oh.
repeat it again.
So I'm asking about the downward sweep of frequencies, like in the FM beds calling. If it's active, right? If it's active, it has some meaning, maybe the meaning like the protraction of whiskers in rats, although it's much faster.
Yeah.
It's a scanning of some domain, and the domain would be the domain of Doppler shifts in the environment.
Yeah.
Could be.
You see, there was a question whether Doppler shift compensation is auditory feedback, or it could be also sensing of the flight movement and things like this. But I did experiments in bats flying in a wind tunnel, and there they compensated according to the speed over ground. And flying in a helium-oxygen mixture, they've compensated according to the sound speed. So I'm rather sure that it's auditory feedback. Was this the question?
The question was about the function. Why do they make this sweep across frequencies? What's the purpose of sweeping across frequencies?
The FM sweep is used like in other bats for localization.
Yeah, but why sweeping?
Also experiments which show they use the FM bats and measure time delays, they measure direction. So it's a bifunctional system. The CF part is for flutter detection and the FM part is for localization.
But why isn't single frequency enough for localization? What's the benefit of sweeping?
Benefit eines FMs for localization.
Yeah, they not only they know there is a fluttering insect, but they also have to localize this insect, and they use the FM sweep during the approach. The amplitude of the FM sweep increases, so you can say it has the function to localize the insect. Flutter detection means detection, and localization is with the FM sweep.
Yes.
This is an amazing array of discoveries over years, and it only compares to man-made systems. Now, in man-made systems where Doppler is used, as part compression and estimation of speed.
Yeah.
Another very important, significant power comes about by comparing the two independent orthogonal measurements, speed by changing range and speed by Doppler. These are two assessments, completely orthogonal of the same feature. Is there any evidence of this being done?
It's very, very difficult. I'm not a neurophysiologist. I cannot say whether there are two processing areas, but that it's used by men, speed is used by man-made. When I found out that there is Doppler shift compensation, I got visited by an underwater Navy researcher, and he got pale and told, "Oh, you discovered that what we just have built in our submarines."
Well, I can't talk about the systems that I know that do it, but they do it underwater and in the air as well.
I would add to that, that when people first discovered that the emissions of bats were FM, and began to talk with engineers, it turned out that that was just about the time that chirp radar was being declassified.
But there's,
Yeah, just go ahead.
if you have an FM Doppler shift, you have a change in the slope. All right, but if you do cross correlation, that's not very easy to do. It's not useful because you get a little reduction in the detection characteristics, but that won't tell you the speed. FM bats detect an echo or receive an echo, whereas the change in the slope, The frequencies that are not in the place they should be for the broadcast slope are treated as absent. And that triggers a different, essentially, it triggers a version of deconvolution, which results in spurious delay echoes. And I don't want to go into the details, but a Gestalt-like perceptual effect occurs that turns the echo off so that they can compartmentalize the range axis by velocity and focus on particular velocity regions.
Yes.
Which is being used. We do not know with bats. We only know that we jumped from the idea directly to putting it in the model rather than doing an experiment. Where it's not so easy to do a Doppler shift in FM signal in real time. That's the problem. There's a way of doing it now, but we're not there yet.
With most FM signals, the instantaneous frequency is 3 to 5 kilohertz wide. So that, 2 or 3 kilohertz Doppler shift is barely detectable.
Okay, Yossi?
Yeah, I have a question that's not related to echolocation, unfortunately. So there are a lot of young students in the crowd, and I wonder if you, for all three of you, if you can say something about what was the best decision during your career and maybe the worst decision also during your career.
I often get asked from young students, which field should I choose? And my answer is, for us, it was rather easy. We were in a time, there were still many things not have been not looked at. The idea of research is you have to find a milk pot where still some cream is in it, where you cannot avoid to go in and have cream on your fingers. Never go to a place where hundreds of hands have been in the milk pot. Look for your own milk pot and you will be successful. That's the answer. Just for me, it was pure luck that I came to echolocation. It had a little bit to do. I was in the Navy before and got a very good training in radar, sonar, and things like this. So that helped a lot. But I think it's not, the decision is to look for a good milk pot.
You want to answer?
Yeah.
Jim, do you want to
Ellen?
respond to that?
I think that's excellent advice, but it's harder and harder to find those still have full milk pots. You want to do something that you enjoy doing, that you have a passion for, and ideally something that is important, that has significance either in neuroscience or behavior or human lives. I too feel that I was lucky to land where I did. And if I have made a mistake in my career, I mean, I'm sure I have many, many mistakes, but the one that I might be most aware of is changing fields, in the sense that when I'd finished my degree, what I considered the cutting edge of neuroscience at that point was very reductionist approaches to understanding the biophysics of neurons and synapses. And the leading lab at that time was Bernard Katz in London. So I went there and got interested in that subject too. So the rest of my career has been sort of schizophrenic between bats and cellular neuroscience. Probably neither one has made kind of progress that would have otherwise. Nevertheless, I've enjoyed both, so that I've been happy with my careers, multiple so far.
Well, first, I don't recall an occasion in my life when a student ever asked me for advice about their future. And that's certainly right. The only thing that was critical to me was I graduated from high school in the university in the state of Illinois. The University of Illinois was $90 plus room and board that's very inexpensive. That's back when governments took responsibility for educating people. That's different now. I went there and majored in engineering physics, 1 degree, 1 year, that was more than enough. And I bombed out, went to a junior college in New Jersey and found some really good teachers and read Hebb's book. And that got me interested in this. That's all. So it's random walk stuff.
I think another good decision in my life was when I started with doing work in the lab. And I had the feeling I know everything about bats. And then I went out in the field and I found out I knew nothing. I think if you have a sensory system or some kind of information processing, you have, the more you know, the more you can compare, get as much information about different species and so on. And this conference, again, was a good example. I learned so much, not from bats, but from other animals. So the big advantage of being a biologist is to have the chance to compare. The comparative approach brings a lot.
Jan.
I have a question to Jim Simmons. You mentioned the Sanderson model, the SCAP model.
Yeah.
And you said that
Einstein
has done some, has given you insights. Has given you insights.
Switch on, you have to switch it on. Yeah.
So you mentioned the Darlene Ketten research that and then you said it changes everything about how the basilar membrane motion may be converted into spikes. And yeah, so I'm curious if you can enlighten us. Yeah, because of course, you know, it's a low pass.
I'll give you 5 sentences for the whole thing, okay? We know in the inner ear, the hair cells respond at the frequency of the stimulus because cochlear microphonic signals work at the frequency of the stimulus.
We can't hear your answer.
Oh, sorry. Neural responses in the auditory nerve. There's no way we're going to be able to
switch it on.
Isn't it on?
Now it's
the red is on. Oh, okay. There's no way auditory nerve responses are going to explain 1 microsecond, much less 10 nanosecond thresholds. Something has to happen in the inner ear. We know that the insertion of the foot plate of the stapes is halfway up the first turn, so sound is capable of going in both directions at once and then reverberating. We know from the work of Christopher Scherer at USC that there's a reflection from the apex of the cochlea back down. So there's several patterns that are moving back and forth in the cochlea. That's going to encode information about time and frequency much finer in auditory nerve responses than would be predicted by a simple bandpass filter model, okay? The problem is we need to know what those are. And then there's another problem. These oscillatory responses which occur between the midbrain and the cortex, midbrain and cortex, midbrain and the cortex. When you have different frequencies, you have the possibility of multiple parallel heterodyne interference devices. So you're making large numbers of potentially different vernier measurements of delay. That could increase in acuity a lot. Third, we need to know much more. We've found that although mammals have electrical synapses in the cochlear nucleus region, we found in bats there are some unusual arrangements. For example, that they may be arranged along incoming basilar membrane fiber, auditory nerve fibers. So there's lots we don't know. And when I meant this is not my problem, I'll be done with this in a year or two. You guys want to work on it, go ahead.
Okay, and then Peter.
Thank you first of all for this perspective on things. And I guess one of the problems with being a scientist is that whenever you think you've come up with a new idea, discovered something new, it's because you haven't spent enough time in a library, and everything was done in the 60s and 70s typically. And given that the three of you at least are partly responsible for a lot of that, you should be the right persons to tell us what are the outstanding big questions in echolocation. Now we can scoop you afterwards then.
It's a challenge for you all. I think what I would like to experience still in my life is somebody who puts an electrode into the cochlea, records the microphonics of behaving animals. Just training or fly, it would be even better that they fly out and you get the ear characteristics, you get the middle ear, you get everything. just recordings of microphonics of behaving animals. I think it's difficult, but it should be possible.
Alan and Jim, what are your suggestions?
I still think one of the biggest puzzles in echolocation is how bats resolve multiple targets simultaneously. And I'm not aware of an explanation for that so far.
We've made several tries and experiments on that question, and it is not answered. So still, no. We need to know how it works, then where the perceptions are, and how it differs in different bats. That's another major problem. How important is the difference between two different species, both of which may use FM echolocation signals? But for example, what's the difference between Philostomus discolor and Aptesicus fuscus and say a free-tailed bat and Myotis myotis? What are the differences for? Or are they not there? We just don't know how to answer the question, ask the question.
Okay, and the very last question from Antonia, and then I think we are all ready to go to the back.
I have again a question that is not connected to bats, surprisingly. So you've been extremely successful in publishing. So I'm wondering, have you experienced a change in the process of the peer review process, the publishing, like publishing a publication back at the time, and now you're still publishing? So what are your thoughts about getting papers out?
Now we cut to the chase.
It's very difficult to say. I think don't waste too much time to get a 98% paper to a 99% paper, then you will lose a lot of data. There are a lot of dead corpses in my cellar, nice things which have not been published. Try to publish everything. That's one thing. And the second thing is, it's a problem with, if you study behavior, you have a problem with statistics. Everybody asks you for significant, and you know it's not relevant. So I think to find the balance between significant and relevant is very difficult.
Well, if you want to get papers published, the first step is to write them. Okay, so if you find yourself in a situation where you're not sure it's ready to write up, you might ask somebody, but at the very least, you write it up and look at it. But the most important thing to remember is to look both ways before you cross the street.
Another thing is always look for a team. To work together with others really helps a lot. You must have people where you can discuss your data on the same level. So I personally was lucky. I always had people around me which were very critical against me. So I was happy today that they didn't show the shade of picture like that, but not a picture like this. I was waiting for something like this. But look for a team and discuss things. That helps also for publications.
Okay.
Thank you all for attending.
And thank you. Thank you to our pioneers. And will you have the last word for the conference?
Yes. So yeah, just thank you everybody for coming. It was wonderful. I enjoyed a lot. I hope you did as well. So dinner now and then dancing. Yeah, well, we won't miss it.
and a big thank to the organizers, of course. Nobody's listening to me. Wait a minute. We want to thank the organizers for all this. So thank you to the organizers. This has been fantastic. Thank you so much.
And I'd like to thank again Talia, who refuses to come in and get thanked. I tried, but let's thank her again.